Gene editing and gene therapy to benefit all. EJW Science and Research News, EJW News Podcast. Hi friends, in this episode I'll introduce my viewers to some of the latest therapies being developed which bring cheerful news to all. We are aware of transplant of heart, kidney, liver, etc. This deals with transplant of genes. I'll try to explain this in very simple and easy to comprehend language. Gene therapy is like transplanting a gene to correct or change the function carried out by it. We know that the genes are carrying different characteristics of all living things, plants and animals, including man, by producing specific proteins which express the characteristics as we see or identify them. Experimenting on pea plants in his monastery garden in Austria 150 years ago is abbot Gregor Johann Mendel, the father of genetics, enunciated the principle of heredity. Later, Dr. Thomas and Morgan of Johns Hopkins University, US, worked on mutation in fruit fly, Drosophila melanogaster. Now it holds promises for treating a wide range of diseases like cancer, cystic fibrosis, diabetes, heart disease, hemophilia, AIDS, etc. in man. At present, scientists know which genes are producing proteins that develop defective or undesirable conditions. By replacing that specific gene by various techniques, they can rectify that particular condition or disease. Now, gene editing drugs that could correct DNAs are being tested in humans. A patient in New Zealand received gene editing treatment to correct an inherited disorder that causes very high levels of cholesterol. These therapeutics will permanently change a single letter in genetic code at a specific location without making a break in DNA, unlike gene editing CRISPR-Cas9 does. This will avoid a genetic typo, a spelling mistake in nucleotide or nucleic acid, which is the basic structural unit of DNA. This is the latest genome editing technology on human being. What is gene editing or genome editing? Gene editing is the emerging molecular biology technique which makes very specific targeted changes by insertion, deletion, or substitution, just by taking a part of the gene and replacing it with the desired genes. So gene editing or CRISPR cluster regulatory interspace short palindromic repeats, zinc finger nuclease, TALEN, transcription activator-like effector nuclease, oligonucleotide directed mutagenesis. What is gene therapy? How does it work? Therapy is a therapeutic delivery of nucleic acid polymers into patient cells as a drug to treat a disease by replacing defective gene by an effective one. Clinical research using somatic gene therapy has been conducted with several diseases including chronic lymphatic leukemia (CLL), Parkinson's disease, sex-linked, that is X chromosome linked SCID, that is severe combined immunodeficiency, which is caused by mutation in different genes involved in development and function of infection-fighting immune cells. Infants with SCID appear healthy at birth but are highly susceptible to severe infections. What is CRISPR? CRISPR is a prominent genetic engineering technique by which genomes of living organisms may be modified. It is based on a single version of bacterial CRISPR-Cas9 antiviral immune system. Cas9 is a protein which has a vital role in immunological defense of certain bacteria against DNA virus and plasmids. A genetic structure in a cell that can replicate independently of the chromosomes, typically a small nuclear DNA strand in the cytoplasm, and is greatly used in genetic engineering applications. Other common gene editing techniques are zinc finger nuclease, TALEN, and oligonucleotide directed mutagenesis. This is not an easy subject to understand. I have tried my best to make it very simple for you to understand. Please like my share, subscribe my channel. I will soon come with more great scientific research work explained in simple language. Thank you very much for watching and supporting me. I am Edwin Williams, EJW Science and Research News, EJW News Podcasts.